And then I saw Planthoppers, and like, they have these, horns at the top of their head, they shoot wax out their butts, they like use substrate-borne communication, and nobody studies them. And I'm like, I am here, I'm doing this. And so within like 4 months, I decided I'm going to spend the next 40 years, I'm going to get my doctorate, and I'm going to spend the next 40, 50 years, if I'm lucky, studying planthoppers. Planthoppers are pretty cool because, for a lot of reasons, but like aphids and other insects that feed on plant sap is nutrient poor. There aren't enough amino acids in it, and these are essentially amino acids that the insect cannot synthesize itself. And so it has co-evolved with obligate bacteria that live inside of it and provision nutrients missing in the plant sap. All insects that have this kind of relationship, we think that it evolved out of a pathogenic type of situation, because insects will actually enclose the bacterium in a cell, like in a cell wall that the insect makes. And so they're called bacteriocytes. In planthoppers and other insects, those bacteriocytes are organized into an organ, or a bacteriome. There's multiple lineages of these bacteria that have different ages. So in the sister group, or the closely related lineage to the planthoppers, the cicadas, the spittlebugs, and the tree and leaf hoppers, they have 2 obligate symbionts that are all kind of housed together in one bacterium, kind of one, sharing 2 rooms adjacently. Planthoppers have each different lineage of symbiont that they've co-evolved with is organized into its own separate organ, its own separate bacterium. And within the planthopper lineage, which is at least 130 million years old and has over 14,000 species, based on light microscopy data from the 1940s, these organs are arranged differently in different lineages of planthoppers. And so they're the only animal known to make a whole organ for each bacterium that infects them. These bacteria have co-evolved so long with the planthoppers that their genomes have eroded away. So you can't plate these things out. People have tried for decades, and they just sit there and they passively take up the nutrients from the planthoppers. They don't have to make things for themselves, which is why you can't plate them out. And so basically it's only that now we can use DNA to detect these things that we're able to study them. We're exploring bacteria in unexplored habitats that only now we can explore much more thoroughly because we're sequencing DNA to detect these things. Just like in the planthoppers, where you can't culture those bacteria, most of the bacteria in armpits, or really anywhere else associated with humans, can't be grown under artificial laboratory conditions. We're amplifying the same gene in the armpit bacteria that I'm amplifying in my planthoppers. How do you connect this to what people care about? Because people think of bacteria as germs, and the insects are cool and it makes sense in terms of how they live, and like little kids like bugs, so that works. The engaging part, and the part that's been really fun for me that's completely different than the insect work, is the health component and the smelly armpits and belly buttons. I mean, kids love that, and clearly there's, adults understand the relevance to human health. But what's been neat about that is the education department at the museum has been going strong for decades, and they deliver a lot of programs to schools. And at the end of last year, there was this impetus to, connect research going on in the NRC to schools through this distance education program around the state. What we do is we send swabs out to the schools so that teachers or their principal could swab themselves. They mail them back to us, and we grow them out. We talk about, how do you know about the microbes that live on you? Are they all bad? That's not really the case. A lot are beneficial. How do we know through traditional methods of culturing? But also, how do we know a lot more these days about bacteria that live in association with us, and that's through DNA sequencing. But the kids love to see what grows on their teacher. There were 3 different teachers who each swabbed themselves, and I never know if they wanted to remain anonymous or not, and so I put them in alphabetical order, so they knew who they were, and if they wanted to fess up, they could. And the first one that I put out, he had hardly any growth, nothing really. The second one, just like a couple little colonies. And then the third one, in my head, it was like the gym teacher or something. It was just this big, like, a lot of growth. And I can see them through this cart, so I can see the class. And this teacher stands up, yay, because it's him. The kids go nuts and cheer. I'm like, we can send that to you to put on your Christmas card. So it's cool because it's very engaging. It's so important that this is, engagement is obviously very important. But as a scientist, the scientific publication is very important. And to be able to maintain the credibility as a scientist and be useful at the same time is what Rob's group has done, like, wonderfully, and what I, as a more junior scientist, what I want to learn. And so that we're taking the results of original, published, peer-reviewed research and connecting it to classrooms throughout North Carolina and likely beyond. And then that's when legislature and taxpayers say, what is the relevance of this genomics lab that studies evolution that's getting our taxpayer money? Well, that's the relevance. We're really teaching stuff and we're connecting our research to these kids.